All right, so let's go and get started. A couple announcements as always to begin with. First off, we're going to be lecturing today. We will be lecturing on Wednesday. We're going to kind of finish up the semester talking about antibiotics and vaccines, so a little bit more focused on disease treatment, disease prevention then. With this as well, we're going to have then our final Brightspace quiz on then this Wednesday after class. So it'll be same format as the other Brightspace quiz. It'll open up Wednesday after class. It'll stay open until Thursday night. We then are going to have the fourth exam on Friday. The fourth exam is going to cover this last disease unit then. And so that will be on Friday. Same format as the previous exams with a mixture of the multiple choice questions, maybe some matching questions, pathogen of interest questions, and probably a short essay question or two on that then as well. Monday, we'll have a review session for the cumulative final. So Monday, May 11th, that's technically the last day of lectures. But that is just an optional review session. I will be in here at 10:30 a.m. during our normal class time. So just come with questions that you have, things you want me to talk about content-wise as a review for the final exam, which is on Wednesday, May 13th. And remember, the final exam is cumulative. It's 40 questions. It could be anything this semester, 10 questions over any of the pathogens of interest this semester. And I have put all the pathogen of interest slides for exam 4, but also I did put all of the pathogen of interest slides into a single document on Brightspace as well under the study guides module. Questions about any of that? Okay. Today, then, we're going to be talking about antibiotics. So we had left off covering then autoimmunity. Any questions about the autoimmune lecture set before I move on to antibiotics then? Yeah. Mm-hmm. Yeah, I feel like I've already been fairly clear on that. I've said several times throughout that autoimmune lecture what I would expect you guys to know. So for instance, if an autoimmune disorder is organ specific, you should know that, but also what specifically organ does it impact then? So for instance, something like Hashimoto's thyroiditis is going to impact the thyroid. For instance, Addison disease is going to be the adrenal glands. If it is systemic, you should know it's more systemic. In some cases, you might want to know the underlying causes. Is it self-reactive antibodies causing it, or is it self-reactive T cells that cause it? So for instance, something like, rheumatoid arthritis is caused by self-reactive antibodies against other antibodies that then form immune complexes, which then activate complement and inflammation of the joints. Or something like multiple sclerosis is caused by self-reactive T cells. Okay. Yeah, other questions? Okay, today we're going to talk about antibiotics because this is a big topic in healthcare. And also, there's a lot of patients that use antibiotics incorrectly, and it's really important to understand what antibiotics are and why it's an issue if they're not being used correctly or properly then as well. Antibiotics were actually discovered in 1928 by Alexander Fleming. So Alexander Fleming, he was a microbiologist. He was actually studying Staphylococcus aureus, which causes staph infections. So he was studying a bacterium, Staphylococcus aureus. Now, Fleming was working with it, and what he noticed one day is that he had gotten a contaminant on one of the plates. And it was just something that got a contaminant on there, wasn't supposed to be there. But what he noticed was this, that there was a contaminant, and this would be the contaminant colony on there, but next to the contaminant colony, the bacteria wouldn't grow up. And what it did is there was this area kind of referred to as a zone of inhibition where there was this fungal colony. And in this case, the fungus was a fungus called Penicillium notatum, which is notable because it makes penicillin. That's where it gets its name from. But he noticed this fungus was actually inhibiting the growth of this bacterial species. Now, Fleming went on. He thought this was pretty important. He thought he found something that could kill and treat staph infections, for instance. He went on to characterize penicillin. He then promptly abandoned the project because he couldn't figure out how to mass produce the penicillin in any kind of notable quantity. It was actually until a couple chemists came in around about a decade later that they figured out how to actually purify the penicillin and get enough of it to, say, treat disease. And then penicillin started to be used in the early 1940s. It really started to be used around 1942 at the tail end of World War II to start treat then bacterial infections. Now, antibiotics, what are they? There are some microorganisms that will synthesize and secrete chemicals that are then going to inhibit or stop the growth of other microorganisms. So I might have, for instance, this might be a bacteria or a fungus. And what it does, it's also an antibiotic producer. So it makes these antibiotics, it's secreting out those antibiotics to its surroundings, and then these antibiotics are killing other microorganisms, other bacteria. And so these chemicals that are killing the bacteria, those are referred to as antibiotics. And antibiotics, by the way, they kill bacteria. Basically, this is what they do. They're chemicals that kill bacteria, and specifically bacteria. Okay, so they kill bacteria and only bacteria. Why would some type of bacteria or fungus be making antibiotics to kill other microorganisms? Why would they do this? So this particular species might be making an antibiotic. It's killing other microbial species. What's the benefit of that? Yeah, it's trying to kill its competitors. So for instance, this is a plate. It's a very dirty plate with lots of stuff growing on it, all over it. But occasionally you see something like this colony and there's a clearing around it. There's no other microorganisms growing near it. That lets it take advantage of all the nutrients on that particular part of the plate. It's killing its competitors, which is then going to make sure that it gets more food and it's more successful then. And this is very common in a lot of, especially bacteria and fungi that grow out in the soil. A lot of soil bacteria, a lot of soil fungi are antibiotic producers. Antibiotics is not something that humans invented. We didn't come up with these. We didn't design these. What we've done is we found a microorganism makes an antibiotic, like, for instance, Penicillium notatum fungus makes penicillin, or Streptomyces bacteria makes kanamycin or chloramphenicol. And then what we've done is we found these microorganisms already making these chemicals, identified those chemicals, purified those chemicals, figured out how to mass produce them, but then we can use them to treat disease then as well. But almost every single antibiotic comes from studying microorganisms. It's not human invented. I think the one example of that is triple sulfa drugs, which also tend to cause allergies more often than other kinds of antibiotics. But that's the whole one, the class of antibiotics that is human designed. Antibiotics are typically fall into one of two categories. They can be bacteriostatic or bactericidal. Anybody know what the cidal term means? Killing. So cidal is just like homicidal, things like that is going to be killing, okay? Bacteriostatic antibiotics, they don't kill the bacteria, but they stop its growth. So when I saw static, they just mean it's stationary, it's not growing. So they suspend the growth of bacteria while the antibiotic is present. Bactericidal antibiotics, these are going to be types of antibiotics that actively kill the cells upon exposure to those antibiotics. This right here is an example of what is called a Kirby-Bauer assay. This is basically where you just take a bacteria and you plate what's called a lawn of bacteria over the plate. You put the bacteria all over the plate, okay, and completely cover it. And what's supposed to happen if nothing else occurs is bacteria is going to grow everywhere. And this is bacteria just growing everywhere. But you apply the bacteria to the plate. Before they grow up, you put these antibiotic discs on the plate. So this is going to be discs with different antibiotics then. In this case, this is a disc with one particular antibiotic, but you can see we can get a clearing or a zone of inhibition. That means that this antibiotic is either stopping the growth of bacteria or killing the bacteria. In other cases, though, the bacteria, we don't see zones of inhibition, and that means that this particular bacteria is resistant to those antibiotics. With something like the Kirby-Bauer assay, we can determine if a bacteria is sensitive to an antibiotic, the antibiotic's going to kill it, or if it's resistant to an antibiotic, the antibiotic's not going to kill it. The one thing is, though, this is not going to distinguish between static or cidal antibiotics. This could just be the bacteria is not growing up in this area, or it could be that they're actively dying in this area, but both of those are going to produce this clearing or the zone of inhibition. But this can be useful to determine if antibiotics are going to kill a bacteria or are not going to kill a bacteria. Now, antibiotics, we use antibiotics that show what is called selective toxicity. Humans can use antibiotics in order to treat and kill bacterial infections because antibiotics are selectively toxic. Selectively toxic means that they prevent the growth of some organisms, in this case for antibiotics, bacteria, but they permit the growth of other types of cells, in this case, human cells. So the antibiotics are going to target bacteria, but not human cells. And that's why we can use them to treat disease and active infections in bodies then. So I think I've asked this question before. Could you use, say, bleach to treat an infection in somebody's body? No, because bleach is not selectively toxic. Bleach is just toxic. Okay, bleach is going to kill everything it comes in contact with. And so if you're using bleach on a person, you're actually killing a lot of their cells. Too much bleach, gonna be serious issues and or death then, for instance. Antibiotics are useful to treat disease because they are selectively toxic. They kill the bacterial cells, but not the human cells. And that's why we can use to treat them. To be selective, though, the antibiotic must target specialized functions in the cell. So when we're talking about selectively toxic antibiotics, the antibiotics, they have to do a couple things. They have to target something that's important to the bacteria. So antibiotics have to target a system or a process the bacteria require. Something important to the bacteria, something they need to grow. Okay? And if you disrupt that system, disrupt that process, you disrupt the bacterial cell. Okay? They also have to target proteins or pathways that human cells do not have or do not use. Or alternately, to be, target enzymes for which humans have a similar but substantially different version of. So it has to be targeting a difference between bacterial cells and human cells. Something the bacteria have, human cells do not. And something that's also then, point one, important to the bacteria. And a lot of complex multi-step processes provide quite a few targets then. What's something that then bacterial cells have that human cells don't? Sorry, what was that? Yeah. So remember, bacteria have a cell wall. What's a cell wall made out of in bacteria? Peptidoglycan. Do human cells have a cell wall? No, There's no cell wall around there's no cell wall around animal cells in general, all animals. Okay, so all animal cells don't have a cell wall around them. But that can be a protein or pathway or target that, again, human cells do not use cell wall, but as is important to the bacterial cell. And they also can do this. So actually, cell wall is a very common antibiotic target. So there are several different antibiotics that will target peptidoglycan cell wall, but also peptidoglycan synthesis, making new peptidoglycan cell wall. For instance, penicillin targets then these amino acid cross-linkers that are in the cell wall structure. And they make it so that you cannot cross-link those amino acid cross-linkers. You can't make new cell wall. Vancomycin makes it so you can't make the polysaccharide chains that are in a peptidoglycan structure. Bacitracin. Anybody ever heard of bacitracin before? You should have if you've been paying attention. It's pretty much what's in almost all antibiotic creams you might buy over the counter in the store and put on cuts and stuff like that. It's usually bacitracin in there. Bacitracin also inhibits peptidoglycan synthesis. There's this little part here that goes to go from the inside of the cell to the outside of the cell, and it inhibits that transport of that sugar unit across the cell, so you can't make new cell wall, basically. But there's several different antibiotics that target peptidoglycan or making new peptidoglycan, and you can target it at different steps in its synthesis. And so inhibition of cell wall is a common route that many antibiotics target. And as you target the cell wall, that peptidoglycan will weaken, and eventually it can cause lysis of the bacterial cell. So this is going to be some bacterial cells before penicillin treatment versus after penicillin treatment. It weakens the cell wall until it gets disrupted, and eventually that can actually break open the cell as well. Another common target is ribosomes. There are several antibiotics that target ribosomes. For instance, tetracycline will target tRNA entry into ribosomes. Chloramphenicol blocks joining together of amino acids and peptide bond formation. Erythromycin blocks a ribosome translocation so that the ribosome can't move along a messenger RNA molecule, read the messenger RNA, and make a protein. So there's several different antibiotics that target translation and are translational inhibitors. So this is several different of these. With that being said, do both bacteria and human cells have ribosomes? Yes. So how is it that these antibiotics then are targeting bacteria but not human cells? Anybody remember what kind of ribosomes bacteria have? And then eukaryotes have what? ADS ribosomes. It turns out there are structural differences in the ribosomes of a bacteria versus eukaryotic ribosomes like our ribosomes as eukaryotes. The ADS ribosomes are bigger, have a few extra parts, have some structural differences. And these antibiotics target 70S ribosomes, but not ADS ribosomes is the idea here. So there is a structural difference between those two different types of ribosomes, and the antibiotics specifically only bind to and target the 70S variety of ribosomes and just target then the bacterial ones. Now, I'm going to bring up, this isn't a pathogen of interest, but an important species of bacteria, an important genus of bacteria is the Streptomyces bacterial genus. Streptomyces bacteria, these are gram-positive soil bacteria. Initially, they were thought to be fungus. They form these little fuzzy colonies. And by the way, if you see fuzzy colonies on a plate, that's usually some type of fungus. Not always. But that is because fungus usually grow in these long filamentous chains of cells called hyphae. It turns out streptomyces also grows in these long multicellular hyphae forms. So initially it was mistaken as a fungus, but then once they started studying it more, they realized, no, it's not a fungus, it's a bacteria. For instance, it has peptidoglycan cell walls, it has bacterial 16S RNA, it's a prokaryote, not a eukaryote, and things like that. So it's actually a bacteria, not a fungus. The reason I bring this up is a lot of bacteria, a lot of antibiotics we know of are actually made by some type of bacteria in the Streptomyces genus. So when these bacteria are growing, they grow in these filamentous chains of cells, but during starvation, it'll start secreting antibiotics. And again, what it's trying to do is kill its competitors. Okay, and so it makes these antibiotics try to kill these bacteria, these competitors of it, and then that will help it feed better. Now, with Streptomyces, there's several different species of Streptomyces, like Streptomyces avermatilis. Avermatilis makes an antibiotic called avermectin. There's Streptomyces SF1293, which makes Biolefus. There is Streptomyces verticillus that makes bleomycin. Streptomyces is a species that make globomycin. Streptomyces canamydoceticus makes kanamycin. There's one that makes chloramphenicol, streptomycin, erythromycin, vancomycin. These might be some antibiotics that you guys might have heard of before. Some of those might be even ones you might have taken before in treatment of a disease. But all of these come from some type of bacteria that falls into the Streptomyces genus then. And about 75% of all the world's antibiotics we know of comes from some type of Streptomyces species. Many of these species make unique antibiotics, and in many cases, they also make multiple different antibiotics. Now, these are growing out in the soil. They're using the antibiotics to kill their competitors. We've discovered these, characterized these, and co-opted these antibiotics then for treatment of disease in humans, though. Now, these antibiotics, by and large, are actually modified fatty acids. Very complex fatty acids, but modified fatty acids in many cases. This is the structure of erythromycin versus globomycin versus vancomycin. They look very different one another, but structurally and how you make them, they're actually fairly similar to one another. So how cells synthesize these, make these, is just some modified enzymatic reactions. And you can get all these different kinds of antibiotics made. Now, what happens is these are bacteria or fungi that are antibiotic producers. So somewhere in their genome, let's just say this is a snapshot of somewhere in their genome, somewhere in their DNA sequences, they're going to have what is called a synthase gene. These synthase genes are actually some of the largest known genes. They're huge. A gene, remember, is a segment of DNA that codes for a functional protein. And by the way, if you guys haven't realized this, I've asked for the definition of a gene on every single exam so far this semester. Anybody notice that yet? Okay. Also, that's a hint. If it's been on every previous exam, okay. Synthase genes are really big genes. They can be about 25,000 base pairs or nucleotides long, 25,000 ATCs and Gs. Again, a gene is a segment of DNA that codes for a final functional product. In this case, these genes, if this is my gene right here, it's a really big gene. It's going to be an antibiotic synthesis gene. And these genes are going to be encoding enzymes that then produce the antibiotic. But I might have a bacteria making an antibiotic that kills bacteria. problem with that? Yeah, why is the bacteria not killing itself? Streptomyces are bacteria. They're making antibiotics. What do antibiotics do? They kill bacteria, period, done. There's no differentiation. They just kill bacteria. So why is the bacteria that's making an antibiotic, why is it not killing itself? Yeah, that's where the resistance genes come into play. There's also going to be some genetic regulators that control when the antibiotic is produced or not. Don't worry about that too much. But there's also then going to be some genes over here. This is going to be a series of genes as well. And those genes are going to encode enzymes that give resistance to that antibiotic. So these antibiotic producers, they have to have antibiotic synthesis genes, but they also have different genes that codes a different enzyme that will let them be resistant to their own antibiotic then. Okay, so there's genes that provide resistance for the antibiotic that is being made. Now, how is it that these are going to then be resistant? What are those gene products or those resistance genes? What are they coding for? And how are they conferring resistance? Well, one way to do resistance is just what is called exclusion. Don't let the antibiotic in. Okay, so one way that bacterial cells can be resistant to antibiotics, don't let the antibiotics in. So you prevent the antibiotic from entering. So for instance, something like, say, let's just say my pink triangles here are penicillin. This is not membrane soluble. What that just means is penicillin cannot pass through membranes. Penicillin hits a membrane, bounces off of it. Okay, what does penicillin target, though? Targets the cell wall. Mentioned that earlier. Penicillin will target the cell wall. In this case, with gram-negative, remember with gram-negative, they've got an outer membrane, they've got a cell wall, they've got a plasma membrane. Okay, so with something like my gram-negative bacteria here, it's got the it's got the cytoplasm, plasma membrane, cell wall, then the outer membrane outside of that. In this case, though, many gram-negative bacteria have that outer membrane. Penicillin comes, hits the outer membrane, just bounces off. It never reaches the cell wall in the first place. As it turns out, penicillin can only kill gram-positive bacteria because their cell wall is on the outside. They don't have gram-positives, don't have an outer membrane. Okay, penicillin will kill gram-positive bacteria. Penicillin cannot kill gram-negative bacteria, and this is an exclusion strategy. Another exclusion strategy, though, are what are called antibiotic pumps. This is where the cells actively pump the antibiotic out. In this case, maybe my antibiotic gets inside of the cell. Maybe in this case, my antibiotic is tetracycline. But there are some species of bacteria that have a gene called TET-A, which codes for a tetracycline exporter. It's an enzyme that any tetracycline that gets into the cell, it binds that tetracycline and then pumps it right back out of the cell. Tetracycline was one I mentioned earlier. It targets ribosomes. So tetracycline gets into the cell, stops ribosomes from working, stops protein synthesis. But this exporter pumps any of that tetracycline right back out, so it never has time to target the ribosomes, and then it's resistant to the antibiotic. That's another exclusion strategy, these antibiotic pumps. Another way that some bacteria are then resistant to antibiotics is they inactivate the antibiotics. So when I say inactivation as another strategy, that is they're going to chemically inactivate the antibiotic in some way. So for instance, sometimes they enzymatically destroy the antibiotic. So there's going to be an enzyme gene that codes an enzyme that is then going to destroy the antibiotic. So for instance, there is an enzyme called beta-lactamase. Beta-lactamase is going to be the one in actually kind of green here, is the beta-lactamase. So this is my beta-lactamase. It gets secreted out by the cell, but then it binds to penicillin and dices it up into fragments. So the beta-lactamase enzyme destroys the penicillin before the penicillin can reach the cell, before the penicillin can target the cell. That can be a way that these bacterial cells get resistance as well. Another way they can do this is instead of just destroying the antibiotic, they can also enzymatically modify the antibiotic. So for instance, chloramphenicol acetyltransferase. Again, most biological terms can be read backwards and they define themselves. I mean, it takes a little bit more than that. What's the ASE ending mean? Enzyme that transfers an acetyl group onto chloramphenicol. That's just what that name means. You read it backwards, it tells you exactly what it's doing. It's an enzyme that transfers an acetyl group, a 2-carbon group, onto chloramphenicol. But when they add the acetyl group onto it, and this is supposed to be my acetyl group coming on to chloramphenicol in pink, that inactivates the antibiotic so it can't function properly. By just adding this chemical group, it interferes with the function of the chloramphenicol. Chloramphenicol is now the bacterial cell is resistant to it. Questions about that? Okay. Another one is just be immune. Usually this is going to be a spontaneous mutation in the antibiotic target. So for instance, streptomycin is an antibiotic that binds to ribosomes, and then it stops protein synthesis. But there are Streptomyces species where their ribosomes have mutated in such a way, changed the structure in such a way that the ribosomes are still functional, but streptomycin can no longer bind to it. So this is a modification and a mutation of the cellular target of the antibiotic then. But Streptomyces bacteria are typically always resistant to streptomycin antibiotic because of that. All right, questions about those resistance strategies? All right, why do I bring that up? All of this requires a resistance gene of some kind. All of those resistance strategies require something that's genetically encoded. The thing about that is then, again, just as a summary, bacteria make antibiotics that can kill other bacteria. But antibiotic-producing bacteria contain genes that then are also going to encode resistance to their own antibiotics. But I also talked about the fact that bacteria sometimes like to do horizontal gene transfer. And we talked about things like transformation, conjugation, transduction. Earlier in the semester, those 3 different types of horizontal gene transfer. And sometimes what gets transferred is resistance genes. In many cases, those antibiotic resistance genes get passed to other types of bacteria species by those types of horizontal genetic transfer mechanisms. And then those resistance genes get introduced to species that were not previously resistant. In particular, a lot of resistance genes are also found on plasmids, those small little circular pieces of DNA molecules. So you might have an antibiotic producer, has resistance genes in it, but somehow that makes those resistance genes by horizontal gene transfer end up in a resistant pathogen. Now you've got an antibiotic resistant pathogen. So you actually, how does resistance spread? Again, a main mechanism is you get resistance genes passed via horizontal gene transfer, but also you just have evolution and natural selection. There's also just evolution, natural selection, evolution. This is also going to favor resistant strains anytime antibiotics are present, especially if there is high levels of antibiotics around. Any type of environment where there's a lot of antibiotics around, you tend to, over time, have resistance evolve in those populations, and then natural selection will favor any of the strains that are resistant, and they become the most common variety of the bacteria around. Then also, they have resistance genes that can also get passed to other bacteria. So it's kind of almost a feedback loop then as well. This is seen again and again. The more an area uses antibiotics, the more likely you have resistance. So here, this is a graph of total antibiotic usage by country versus then penicillin non-susceptible or penicillin resistant Streptococcus pneumoniae infections. And as used, general trend, as you use more antibiotics, you get more resistance. Okay. The more antibiotics are around, the more faster evolution will favor antibiotic resisting strains. This is also showing the spread of antibiotic resistance in several different categories of antibiotics, like fluoroquinolones, carbapenems, cephalosporins, aminoglycosides, polymyxins. Don't worry about that, but there's different kinds of antibiotics that fall into different categories or groups. And this is resistance that kind of was developing in the early part of the 2000s to several of these. By the way, this problem's gotten worse. I need to find a more updated data. But in some cases, you're finding 20 to 30% of all bacterial infections are resistant to antibiotic treatment. Oops. Oh. Including, there are some strains, which I have asterisked here, that are these strains, subtypes of things like Enterococcus faecalis, Pseudomonas aeruginosa, Mycobacterium tuberculosis, which cause TB. There are some strains out there that are now multi-drug resistant and actually are resistant to and untreatable by any of the antibiotics that we currently have to treat disease. So for instance, somebody in Wisconsin in the early 2000s had a multi-drug resistant version of tuberculosis. They put her in the hospital in quarantine for the rest of her life, because her version of TB did not respond to any antibiotics. There was no cure for it whatsoever. They put her in an isolation unit until she passed away from the tuberculosis, because they could not have that escaping out into the general population. So how do we actually then prevent resistant pathogens? There is things we can do to limit the spread of antibiotic resistance in bacteria. And by the way, antibiotic resistance is really bad. Do you guys, pretty much any surgery, anybody that gets a surgery done is put on prophylactic antibiotics. You guys know what prophylactic means? Just in case. Preventative antibiotics. They don't know if they have a bacterial infection. We're just going to give them antibiotics to prevent any bacterial infection, just in case. Okay? Does that make sense? Okay. So surgery patients always get a course of antibiotics afterwards to prevent infections. Doesn't always work, unfortunately. That's because there's resistant strains. Resistant strains of bacteria make surgeries much more high risk because you're worried about an infection. And so these resistant strains I think I talked about this at the beginning of the semester. Anybody remember what the number one cause of death in the US is currently? Heart disease, followed by number 2, cancer. Number 3 for a while there was actually COVID-19. I think that's finally fallen off a little bit. I think now it's more like accidents, for instance. But if we lump a bunch of different kinds of accidents together and things like that. But the number 1 cause of death prior to antibiotic and vaccines was infectious disease. So before the invention and the usage of antibiotics, before vaccination. By the way, antibiotics, vaccination, they started getting used about the same time in the 40s and 50s, 1940s, 1950s. But before that, infectious disease was the number 1 cause of death. So antibiotic resistance, very bad. You're talking about now diseases becoming untreatable, surgeries more high risk, a number of different things that's going to be a heavy impact on healthcare and things like that. So how do we prevent resistant strains? Well, first off, again, remember, we could just make new antibiotics. That's great. We come up with new antibiotics that didn't even previously exist. There's not going to be resistance to those yet. The problem is it takes about 20 years to find, develop, and test a new antibiotic and make sure that it is safe for human consumption. They've sped that up sometimes about a decade. Doesn't matter. Even at a decade, it takes only about 2 years of widespread antibiotic use before bacterial populations start to show resistance to it. So that is going to be losing math. Okay. Evolution of bacteria is way faster than what we're going to be able to come up with new antibiotics for. Only take back to antibiotics for bacterial infections. Remember, what do antibiotics kill? Bacteria, and just bacteria. So is an antibiotic going to do anything for a cold or a flu? No, because those are viral infections. Cold is a virus caused by viruses. Flu is caused by flu. COVID-19 is caused by SARS-CoV-2 virus. Antibiotics are not going to do anything for a viral disease. And so, again, remember, the more you use antibiotics, the more likely that you are to get resistance developed. When I was a kid, parents would take their kids in sick. The doctor would say, here's some antibiotics to make the parents feel better. They gave it out like candy. Okay? They don't do that anymore. Now you have to do, oh, you think you have a strep throat infection? What do you have to do before you can get antibiotics? You have to do a strep test, and you have to test positive for the strep test before they'll prescribe the antibiotics. If you do have a doctor that's prescribing antibiotics without doing the test or waiting for the test, they're not doing what they're supposed to be doing, which still happens sometimes. When you're sick, you should take your full prescription of antibiotics. Typically, you're going to usually have to take antibiotics for 10 to 14 days. Some people take their antibiotics for a day or two, start to feel better because the antibiotics are doing their job and the antibiotics are doing what they're supposed to be doing. But then they stop taking the antibiotics. What's the problem with that? Yeah, remember the bacteria are often growing in biofilms. You might have killed the outer layers of the biofilms, and yes, you're killing some of the bacterial species, but only a couple of days of the antibiotics, that's not enough to kill all the bacteria in your body, especially if they're in biofilms. And so then you come off the antibiotics and the bacterial population just pops back and you get sick again. The other problem is you just expose them to antibiotics. And sometimes what ends up happening is the bacteria are going to evolve and change over that time. And the ones that were more resistant to the antibiotics are the ones that are surviving and more likely to pop up. If you take your full prescription of antibiotics, that's sometimes enough to kill them still. Okay? You can take a combination of antibiotics. Okay, and so while some bacteria are resistant to a single antibiotic, sometimes if you hit them hard enough with multiple antibiotics simultaneously, that's still going to kill them. That's too much for them. Their resistance genes aren't sufficient to overcome multiple antibiotics simultaneously. This is just an example of this. This is a Kirby-Bauer assay test. If you have a single antibiotic, this might be the zone of inhibition. If you have this single antibiotic, this might be its zone of inhibition, like this distance right here, right here. But when you have a combination where they overlap, you get more killing. You get a bigger zone where there's an overlap. Again, over this direction is just one antibiotic. Over this one, it's just this antibiotic. But over here, you get these big zones where the two antibiotics overlap, and you get what is called a synergy or a synergistic effect. So sometimes two drugs together are more effective than either one of them alone. Stop using antibiotics on farm animals. About 25 million pounds of antibiotics are used on farm animals per year in the US. Cows, chickens, pigs, things like that. Has nothing to do with treating disease or preventing disease. What ends up happening is, yes, sometimes it's given them to protect from disease, but usually they're given in their daily feed as, in essence, almost acts like similar to steroid hormones, and they grow more muscle. They get more muscle. And so by putting antibiotics into the daily feed, and they don't actually know why for real, but it ends up that you get more muscular cows, more muscular chicken, more muscular pigs, but more muscle equals more meat equals more profit for the farmer. And so what ends up happening, though, is this makes antibiotics very prevalent on farms. And again, the more antibiotics you have around, the more likely resistance is to develop. So by having a lot of antibiotics on farms, you get antibiotic-resistant bacteria. Those resistance genes spread by horizontal gene transfer to other bacteria, including pathogens. Farmer gets sick now with a pathogen that's antibiotic resistant. They go to the doctor, they go to the hospital, they go to the farmer's market, and they're going to start spreading that resistant pathogen to others in the population. Questions about that? By the way, you can get a similar impact by growing like the flocks or the herds in smaller cohorts, where you don't have them in big giant herds with as much competition for food between all of them, and you actually get also more muscular animals if you do a better job of dividing them out and give them. Anyways. You can also put antibiotics on a hiatus. What does hiatus mean? Break. Yeah. Let's just stop using for a while. 5 to 10 years. Whoops. If we stop using an antibiotic temporarily, we find that resistance to that antibiotic will decrease over time. Antibiotic resistance genes are only beneficial if the antibiotic is around. Otherwise, they're a cost with no benefit. So what will tend to happen is if, say, you stop using penicillin for a while, and by the way, penicillin really isn't used at all anymore. Too many bacteria are resistant to it. So they've stopped using antibiotic. That penicillin antibiotic is on a hiatus, but over time it will reduce. No, penicillin is, the allergies to penicillin are for a different reason. Penicillin is one of the few molecules that can actually trigger all 4 types of hypersensitivity allergies. So it can trigger type 1, type 2, type 3, and type 4 hypersensitivities. And it's one of the only things I know of off the top of my head that can trigger all 4 types of allergic reactions. That's why, because different people have different hypersensitivities to it. Most of the time when you say you have an allergy, it's type 1. Type 2, 3, and 4 are some other stuff, but it can trigger all those. Okay. For today, the last thing I want to talk about is carbapenem. Carbapenem. This is an antibiotic that disrupts bacterial cell wall. Its use is restricted and heavily restricted. This is what we consider an antibiotic of last resort. This is never given to people normally. It's given to a patient where the bacterial infection has not responded to any other previous antibiotics, and then they give this in the hope that it's going to work. Okay, so this is used in the case of severe infections that haven't responded to other antibiotics. Okay, so this is supposed to not be used. Unfortunately, you can buy it in the black market in India. Yeah, multiple reporters have actually gone in there with very little background and been able to buy this one in India with no problem whatsoever. Yeah. Last thing for today is then I'm going to go through this real quick. Klebsiella pneumoniae C, or KPC. This is a common gram-negative intestinal bacteria. It can be an opportunistic pathogen. What's that mean? It only typically affects immunocompromised individuals. It is a very common nosocomial infection. What's that mean? Hospital-acquired infection. It can cause pneumonia, bloodstream infections, wound or surgical infections, and in severe cases, meningitis, which is a brain and spinal cord infection. Treatment is antibiotics for Klebsiella pneumonia infections. The problem is with KPC, there is a strain of it that is unresponsive to any antibiotics, including carbapenem. So KPC is a version of Klebsiella pneumoniae that is multi-antibiotic resistant, including the carbapenem. This actually killed about 20 people at the NIH teaching hospital in Bethesda. By the way, this is like one of the hospitals in the US. This is like one of the premier high-tech hospitals in the US. It specifically serves immunocompromised patients and only heavily immunocompromised patients. It's all positive pressure rooms, sterile techniques. It's like super duper training for all the healthcare professionals. KPC started spreading through the population and killed like 20 patients in there. They still don't know how it spread, but because it is immunocompromised people and because it didn't respond to antibiotic treatment, there was nothing they could do. Just deadly bacterial infections in those patients in the absence of effective antibiotic treatment. All right, sorry I went a little over today.